So who decided to go first?
So should I go, Sang, or?
Yeah,
Okay.
go ahead.
Okay. Well, then
All right, I'll do well, I just wanted to thank everyone for coming to our 5th Meet the PI event. This event is our first and only immunology-based seminar, which is kind of special. And so far, we've gotten pretty good feedback. I'm really glad that everyone enjoys our events. I wanted to take this series to really highlight a career path in academia and kind of let all the students that are joining really gain an insight on how a career path in academia starts. And then if we talk about, I like the brutal honesty of kind of like navigating through the career path. And it's also very interesting to hear everyone's experiences because everyone has their own unique experience and gets to where they are now in a completely unique way. So with that being said, moving forward, we're going to hear from Dr. Botond Igyarto. Did I say it correctly?
close enough.
Close enough. I'm sorry if I
No worries.
say it
I
a
got
lot
used to it.
All right, so the floor is all yours. And again, thank you both for agreeing to do this back in September and being so enthusiastic about it. I really appreciate the time and effort you put into this.
Thanks, Gina, for inviting us. I'm really happy to be here. To be honest, I really hate presenting through Zoom because I don't hear the people, I don't see them. So I really hate talking to my screen. So if you want to turn on your microphones, please be my guest. I'm really happy to hear the feedback directly. So that being said, let's go ahead and talk about my lab first of all. And I won't spend too much time on it because I already presented my lab a couple of months ago, but I give you a taste about the Igyarto lab. So what do we do in the Igyarto lab? First of all, besides having fun, we do some research on dendritic cell biology, and we are trying to develop dendritic cell-based vaccine for pretty much everything. It can be cancer, it can be infectious diseases or allogeneic organ transplant. So whichever you want to do. But let's talk about what are the dendritic cells. Well, dendritic cells are, in a layman's term, they are the trainer and the coordinator of the immune system. They are pretty much the boss. They are calling the cards. They are telling everyone what to do and how to do it. So maybe I'm a little bit biased, but if you want to study immune system, I think I would focus on dendritic cells, right? Now, that's about dendritic cells, and let's see what kind of projects we have. I have a pretty diverse lab, and the projects are also really diverse. As I said, they pretty much cover all different aspects of the immune system. The only common thing between these is that the dendritic cells are in the center of attention. And you guys can go into and look at what kind of projects we have. The only other thing that I want to mention to you is that if you ever consider coming to my lab, you may want to focus and read through these so-called lab standards. And if you think that you can associate yourself with these, please come and find me. Otherwise, I don't think that we should waste each other's times. So this might have sounded cocky, but in order to not to go insane, I think there should be standards all the time, and you have to adhere to it. Now, that's about the lab. I am not going to spend any more time. You guys can go into it and read about. Let's spend a little bit of time about my career path. Maybe that's going to be a little bit more interesting to some of you at least. And I really want to introduce myself, telling you where I came from. So this is Europe, of course, and this is Hungary, and this is Romania. And if we move and zoom in a little bit, we will see Transylvania. And this is actually, you probably heard about it from, at least from the crazy Hollywood movies about Dracula. Anyways, so this Transylvania used to belong to Hungary, but after Hungary lost the First World War, became part of Romania. But there are still plenty of Hungarians living there, and I was one of them. I was actually born and raised there. And then I went to school to so-called Kolozsvár, and I did my BS training at University of Bábes-Bolyai. focusing on biology. To be honest, I never finished my MS because I also got into the graduate school in Budapest, Hungary at Semmelweis University. And here, I was involved in immunology. And why did I go in immunology? Is because my father contracted or actually developed an autoimmune disease after a lung infection. And that was the driving force to get me into immunology. And as I told you, dendritic cells are the boss of the immune system. So I started studying dendritic cells and how they regulate the immune system. Now, you have to know that the model organism that we use there, those were actually chicken. Now, after I finished my PhD, I decided that I want to move closer to something that is closer to human and mammalian system. And I decided that I'm going to go overseas and I'm going to end up in United States. Well, I will tell you the story why United States later on, but for now, I just want to tell you that I did a postdoc at University of Minnesota and a lot of other things. As you can see, I spent quite a bit of time there. And after I matured enough to actually wander out and make my own lab, then I moved down to Texas, Dallas, Texas, where I started my lab at Baylor Institute of Immunology Research, where everything was going pretty well. I built up a pretty good lab, but by the time the whole lab was working really well, the leadership decided to move away from basic science to clinical research. So that actually forced my hand to go out and try to find another faculty position. But keep in mind, I did not have papers published and grants at that time. So it was quite challenging for me to secure another faculty position. But I finally ended up here at Jefferson in 2019, where I started my second lab. And to be honest, I really am not planning to start it again from scratch. If I ever have to do it, I will certainly going to do it in Hungary. But the summary of my present standing is done here. I don't want to spend time on it. Let's talk about my journey in United States. And first of all, why United States? Well, even back in 2007 and still today, I think if you want to do science, America is one of the best places to do that. And the second thing was that I really wanted to learn English. And when I got to the United States, I barely spoke any. And then my plan was that I would go back to Hungary after 3 years of postdoc. Well, you might wonder whether did I achieve my goals or not. I certainly did research. That's the first thing. I don't know about my English. I am not going to be the judge, but I certainly didn't go back to Hungary. So I did not really achieve everything that I was planning to. Now, you may wonder why University of Minnesota? Well, that was by accident, because when I signed up to do a postdoc in the United States, my former boss was still at Yale. So I thought that I'm going to Yale. Well, you might ask why that specific lab? Now, again, of course, because of Yale and because of a dendritic cell project. That was a dendritic cell lab, and I really wanted to continue my research in dendritic cells. Alternative that I had was University of Chicago, a T-cell project, an all-Hungarian lab. So you can probably see why I didn't go there, because it wasn't the project that I wanted to work on. And you really won't learn any English in an all-Hungarian lab in the United States, to be honest. So I decided to go with the first option. Now, what did I do in Minnesota? Besides doing science, I did a lot of side projects, and I will present a tiny bit of it just to give you a sense what I was doing besides doing science, and you probably will understand why I'm presenting after I actually tell you. Now, for example, I would buy a car, I would fix them up and sell them. So for example, here I removed the engine, I refurbished it, put it back, and there you go. We bought out the house, pretty much single-handedly remodeled the house. This is just me replacing the whole sidewall. This is the finished product. This is the bathroom before and after. The same thing is for the kitchen. Now, I also did some funny things beside this. Of course, I was growing vegetables, but just to keep the spirits up, I even distilled my own spirits. And then you might be wondering why I am presenting all these to you, right? Well, why did I need side projects? Well, I needed side projects because I had to sustain my family and because handshake is not enough these days to seal the deal. When I signed up for this postdocs project and position, I was promised a standard NIH 38K salary. And when I got to Minnesota, I was presented with a contract with 30K. Now, I did not have an option. I signed up for it, but you can think that this definitely set the mood for the rest of my postdoc. Now, you may wonder, was it a good use of my time? And of course, if you think all the effort and time that I put in these side projects, if I would have put it in my career, by now, probably I would be a full professor. So my answer is no, unless a catastrophe will strike and only self-sufficient people will survive, which is well, maybe highly unlikely, but if you look at the pandemics that are going on around here, maybe it's not that far away, so I can still use my skills one day. But why I'm telling all this to you? I'm telling this to to learn something out of it. So trust but verify, and there is a really good Latin saying, "Verba volant scripta manet," which means spoken words fly away, written words remain. So keep that in mind when you do anything in your life. Now, let me tell you what made me to become a PI in the United States in the first place, because I was planning to go back to Hungary. Well, I had some incentives, and let me tell you about my incentives. One is my wife. She actually joined after my second year of postdoc, and she said, "Look, I've been following you throughout the world. It's time to stop, take a break, and enjoy the American way of life." And she said, "Look, if you get the Nobel Prize, I move with you wherever you want to." So she set the stakes pretty high for me. And the other thing, I got pretty much tired of other people taking credit for my work. And that's why I decided that I will have my own lab here in the United States. But before we get there, the take-home message is that man plans and God, well, if we can consider my wife as a God, laughs, he actually decides what is going to happen. Now, that being said, keep in mind that my mindset was when I came to the United States that I would go back to Hungary after 3 years. And because of that, I never took the time to learn the American academic system. And that was used against me, but I am not blaming anyone. It was my fault. I should have learned it. So at this stage, I actually decided, okay, I will learn this system and I will use it at my benefit. So I did that. But before we continue this story, I want to give you some advice, what you should do in graduate school and even in postdocs, and what you shouldn't do if you want to be successful and you want to have a straight path with careers becoming a PI. Now, what I'm going to tell you is I will divide in graduate school and postdocs. First of all, what you want to do is you find a place, a school that fits your personality. And this is really, really important because you cannot achieve your full potential unless you find a place where you can thrive. I have a lot of other things here that I'm not going through it, but please read it through. It's not a complete list. I'm always open for discussion, all of these things. So come and find me. Now, more importantly, find the school and the PI that is going to dedicate time to mentor you, to get you ready for the next step. And the next step is pretty much becoming a postdoc. So during the graduate school, you have to learn how to design experiments. And here I put a book here, which actually was suggested by my research assistant, Sonia, which is really important. I actually see every day that people are not taught how to do a well-controlled experiment. And unfortunately, that results in you repeating a lot of things over and over. Now, it's highly important for you to remain objective and interpret the data objectively. And even more important is to present your data. Learn how to present your data, how to sell your data and yourself. I see a lot of really brilliant scientists who are really good, but they cannot sell themselves. They cannot sell their data. So they're never going to be as successful as they could. So please allocate time, take classes, take like Toastmaster International, whatever works for you, but make sure that you are a good presenter. You can sell yourself and your data. Another way of actually making sure that you are selling your science is that you know how to write a good manuscript, how to capture, how to sell everything that you produced in the lab. And the next step is that you have to learn how to write grants. Without grants, you're not gonna have money, you're not gonna succeed. So all this is all about marketing. Keep that in mind. Now, if we go further and we wanna talk about don'ts, this is pretty much the opposite that I listed on the left, so I'm not going into the details. What about postdocs? It's pretty much the same idea. Find the right place for your personality and make sure that you research your mentor, because again, that's gonna decide whether you're gonna have a successful career or not. And the good PI is the one who is going to teach you how to secure a faculty job and become and remain successful. Helps you to secure grants that is going to aid your transition to the faculty position. It actually promotes you. They have to list you as a corresponding author on the first author publications that you have. They actually let you to present at invited lectures. That's something that's a really big deal if the PI is invited and he or she sends you to present instead, her or him. Now, more importantly, if you want to become a PI, you have to become a businessman. A businessman who actually running a business without a product to sell. The only thing that you have is your data. So you better become a really good marketer of your data and sell it in order to get funding. Now, you also have to study marketing, as I said. You have to study or actually learn how to do a proper time management, and you have to learn how to supervise people with different social cultural background. Unless you do, some PIs do. For example, if I am from Hungary, I would take only Hungarian students, then I would not have to deal with any of the social cultural background differences. But as you saw, my lab is quite diverse, so I do have to deal with that every day. And I would highly suggest to study human behavior and human psychology, which is again is quite helpful if you are a PI. Again, don't the opposite to the left. That's pretty much what I wanted to say to guide you. Here, the next step that I want to tell you about is how I see the American academic system. And again, this is subjective. Everything that I say is subjective, even though I try to stay objective. It's from my perspective, my experience. So take it with a grain of salt, but I have the best intention to guide you on the path of academic system. So I would suggest to you to think about the American academic system as like an elite club of like men's club, golf club, or whatever club that you can think. So you have to reach certain requirements to get into this elite club. And when you do that, all the doors going to open. I mean, your life is going to be set forever. You won't have problems. You're going to be successful. So here I am just telling you how I would play the game if I would have the chance to start again the whole process. So again, this is from my perspective, and this is the ideal situation, not that it pretty much ever happens. Now, what I would do, I would get the best undergraduate education possible. Get involved in research early. It's highly, highly important to develop your skills and build your CV. I am not criticizing the American system here, but you have to do the check marks on your CV. You volunteered, you achieved this award, X, Y, Z, or whatever. I really don't care about these things when I interview people, but you have to play the game. Now, what you want to do is next to find a highly successful PI in training graduate students and get in his, her lab and do a PhD. And you have to find a mentor who is going to dedicate time to nurture you, to get you ready for the next step, which is becoming a postdoc. And I'm telling you, if you do it in a really good lab, all the doors going to open for you to do postdoc again with a big shot PI, who will support your guest for the faculty position. So you can see it's like a closed society. You have PIs training graduate student, that's going to hand it over to somebody who is going to do it, train you as a postdoc, and he's going to hand you to a different university where you're going to become a PI. Don't do it in a way I did it. I actually joined a fresh assistant professor's lab, and I'm telling you, it's not a good match because you are competing with that PI. That PI has to establish himself. So he or she is going to take everything from you possible to succeed. Otherwise, for them, the tenure track clock is ticking. And if they don't establish themselves, they're going to fail and they're going to lose their lab. One more thing that I would highly emphasize, these are not really bad things. I'm just telling you advance to keep in mind and maybe compensate if you are in that situation. The so-called in-house breeding, that means that you do graduate student postdoc and become faculty at the same place, or you do postdoc and become faculty in the same place. It's not a bad thing, but keep these two things in mind. When you apply for grants, it might be challenging to prove that you are independent from your PI. Some people could also consider you less vetted. What does it mean? It means that you played safe. You stayed in one place and you never went out, you never challenged yourself to try to sell yourself to a different university. But all these minimal things can be compensated for. I'm just putting this out there for you to learn. Now, that's another interesting topic that I want to bring up to you is, well, you might wonder what the Greek mythology has anything to do with scientists. And I tell you just in a bit what it has to do with scientists. And this started out with a conversation that I had with one of my graduate students, Chris Herbst, who actually asked me, Botond, what keeps you in science? What drives you? What makes you to do science every day? So on the next slide, I will tell you what. So this guy, Albert Szent-Györgyi, he was another Hungarian scientist, but he actually got the Nobel Prize for isolating vitamin C and describing the different steps of the citrate cycle. And he divided the scientists in Dionysians and Apollonians. And here I highlighted in red what Apollonians tends to develop established line to perfection. Now, look around in your institute, in your department, and you will see that majority of the PIs or labs, they are highly focused on doing a certain really small slice of the science. For example, they look at the pathway, they look one single molecule, and they study that from all different perspectives. So they are pretty much a master of their field. And that's what, well, unfortunately, NIH rewards that more than anything else. So they want to see when you apply for grants that you are really an expert. You show continuity. And that if you do that, they're going to fund you forever. Now, I don't consider myself in this category. I'm more like a Dionysians, which tend to look in different direction and find unexpected alleys for research. So again, neither of these are bad, but if you belong to a Dionysian category, make sure that you have to compensate and do extra steps in order to get funding. So maybe 5 years from now, I won't have fundings because I'm not as focused than other people. Okay, so that being said, let's talk about so-called universal law of success. And believe it or not, that exists. So I want to share that with you, and hopefully that's going to help you to think about this and use it to your benefit. I'm going to share the work of another Hungarian. This guy actually was born nearby my place. He's a highly successful network scientist. He's at Harvard. He has like 250,000 citations, just for you to gauge it. But he's also a phenomenal marketer of his work. And here pretty much is the takeaway from his book. First of all, you have to be highly productive. So in graduate school and postdoc and in SPI, you have to put out a body of work in public, a large amount that the public is going to understand. So you have to market your research and you have to build relationships. As I said, in the academic system, it's like a closed circuit. You better get out, mingle, know more, learn, and get to know more and more people who is going to help your career. and sell yourself as a person who other people want to keep in their circle. So that's pretty much, it's really basic things, but it's really powerful. So this guy didn't come up with anything new. He just pretty much summarized it and sold it in form of a book. But I think he has made quite important discoveries here. And at the really end, I want to conclude a few things. These are not really conclusions. It's more like sayings that I would tell you to follow during your life. So if you read the first sentence and you are in science and it does not apply to you, I think I would suggest get out of science and find something that you love, something that you will never have to work a day in real life. So find your thing. If it's not science, then get out of science. Otherwise, it's going to be really painful to become a graduate student, postdoc, and a PI. So go and find that. Now, another important thing that Barabashi also mentioned, pretty much no pain, no gain. If you are in science, you should be in science 24/7. I'm not saying that you should be in the lab 24/7, but you still have to work quite hard. And even when you get out of the lab, you should be thinking science and how to improve yourself, how to become a better better person, better version of you. Again, get everything in writing, trust but verify. These are important things. Unfortunately, a handshake is not enough. For me, still. And one thing at the really end, this is again a Hungarian saying, it's a direct translation. I would say that smart people learn from other people's mistake, the stupid ones from their own. Okay, this is really direct. I don't want to hurt anybody. But what I want to say that get a great mentor who is going to hold your hand and walk you through the steps that other people took and they failed, and they make you to skip those steps. So you don't have to fail. You don't have to do the same mistakes that other people did. And unfortunately, I am guilty as charged because I never had a great mentor here in the United States who will do that. I actually do consider myself stupid from this perspective because I did everything by my own. I tried it, I failed, I went further. So that's how I acquired the whole experience and whole knowledge that I'm sharing with you. And if you are considering to become a PI, I would suggest read this book, and they actually summarize everything that the PI needs to follow in order to be successful as well. So thank you for your attention. I'm really happy to take questions. Hope I I actually went over my time. So I apologize. Please come and find me, and I'm happy to discuss with you anything that I presented today.
Yeah, thank you for that. I really liked your side projects you were working on. Yeah, so to save time, if anyone has any questions, we'll have a discussion at the end. And then Dr. Kim, if you want to go ahead with your PowerPoint.
Can you see the screen, PowerPoint?
Yes.
Okay.
I think you're on presenter mode. I don't know if you can do it in, if you just,
Good.
Yeah, perfect. Thank you.
All right. Okay, so I'm gonna talk about my experience so far in my academic career. So maybe along the way I can explain what was good, what was not so good, based on my experience, right? So I was born in South Korea, so you may, if you heard this South Korea, you may think of North Korea first, or BTS, or movie Parasite. But anyway, I was born in Seoul, South Korea, and all my families were there too at the time. And I was educated up to the master's degree at Seoul. So, you know, from high school, when I applied for the college, we went through this, SAT kind of test at that time, and I was supposed to choose the major. And the strange thing is you have to choose a major, and then if you don't get in, then you don't have you cannot apply you cannot submit multiple applications. You only apply one. If it works, you get in. If you don't, then you have to study 1 year again and then reapply. So it was a very strange system at the time. So I chose electrical engineering as my first choice. And my second choice was biotechnology. So my first choice didn't work. And the biotechnology was my second choice and got in. But it was really not my favorite because biology was elective course in my high school. I didn't take it. So I have very minimum knowledge about the biology itself. So I had no idea what I was doing at the time, but at the same time, I didn't want to wait for another year to go to college. So I just decided to move on. So because of that, as you can guess, I was not really interested in studying at all. I was really get bored and I was more focused on like doing other extracurricular activities in my college. So first and second year was pretty much nothing interesting to me at all, particularly when I had to take classes for, like calculus and physics and things like that. I just had no idea why I had to study. And I felt that it was not really relevant to me at all. So things changed a little bit after that when I became a third year because, since my major was biotechnology, I was supposed to start taking real biology classes, like, molecular biology, cell biology, immunology. And those, even though I was not really thinking that biology is something I would do, that really intrigued me, mainly because they are talking about what's happening inside of my body. So that just makes me think, oh, this is really relevant to me. And just learning what's going on in my body just makes me think that it's something really interesting. So that's what I thought at the time. So that was the first time I thought maybe I can continue to study biology and learn what's going on in my body. So there were two career paths, but before that, I even thought about becoming an accountant or going to a law school. But after I learned that, biology is kind of interesting to me, I thought about the career path. So the first one is, studying how, what's going on inside of your body can lead to, go to medical school and just learn how to treat people, disease. Or second choice would be go to graduate school to study more. Now, again, in South Korea at the time, the school system was weird. So medical school was not graduate school. So you have to choose the medical school pathway after high school when you go into the college. That means if I wanted to go to medical school, I had to take the SAT again and go through the undergrad one more time, which was not really ideal. So there were some very small number of chances that I could get into the medical school directly. So those opportunities were mainly for like somebody, like for example, in the medical school, they need the people who can draw illustration for the picture of the human body, things like that. So they need somebody who did the art major, for example. So they had the specific needs for recruiting the medical students out of the college graduates. So even though I was not really eligible that much, I applied anyway and it didn't work. But at the same time, while I was thinking about applying for medical school, I thought about going to the graduate school. But then first I decided to go to the master's program to get some more experience in the bench work. But at the same time, I thought maybe I should go to abroad to study. When I was young, I used to play baseball a lot. Let's see, if you are a baseball player, then you may have a dream like, oh, someday I will go to the Major League Baseball, maybe I can play there, right? So I had something like that in my mind at the time. I read all these interesting, research articles in the field, And I thought maybe I can learn from them directly. So for doing that, then I have to go to abroad, right? That's why I decided to apply. So I studied for GRE and then apply for the PhD program in US while I was doing research in the master's program. And I was really lucky, and I got into the PhD program for immunology at Yale University at the time. So once I start the PhD program, of course, I did a rotation like you guys did before, and I chose very big lab. So they're basically, my former PI has 20 postdocs and one student, which was me. So it was really big lab, and I decided to go for it, not because I like it, more like he's a big time player in the immunology, and he has been like Hugh investigator over for 30 years. So he's a big figure in the field. So I thought I could learn a lot from him in general, which I did. But another thing I did was I was really interested in, again, learning what's happening inside of my body from what I learned in the classes. Now, one thing that struck me at the time was that everybody is thinking about in vitro system, right? At the time, signaling pathway was very popular. So T cell receptor signaling, going down and zeta chain activation, phosphorylation, and the JAK7 activation, blah blah blah. They study this, so yeah, it's interesting, but to me, not so much. In my mind, I thought I should be able to use that knowledge to explain what's going on in my body. So that was a little discrepancy, I would say, from what I expected. Then I thought that maybe there should be some kind of spatial and temporal aspect of hypothesis, not just signaling pathway, but how you can explain what's going on inside the body through that sense, when it occurs and where it occurs, things like that. All those things were missing, I thought. So I decided to develop a project taking those points into consideration. So ambitiously, I initiated a very novel project. Fortunately, I was in the big lab, so funding was not a big issue, but it took a long time because I initiated a project nobody had ever done. And I initiated and then screened and made a knockout and found the phenotype. So it took in total 7.5 years for me to leave the lab after finishing. They let me graduate at the 6.5 years, so still longer. But at that time, my classmates all graduate at the end of 6 years. So it was not that late, but again, it took a long time. But good thing was that I published 2 papers, research papers, and 1 review. And one of the paper was a science, which was really nice for me. So that's what I did for the PhD. And then after that, I did a postdoc training at the NYU, focusing on immune responses in the gut. Again, I was interested in immune responses, not like in the lymph node or the spleen, more like on a non-lymphoid tissues in the distal site to see what's going on in the immune system. So that's why I chose the gut as a site of the focus for the study. And it took 7 years to finish the project. It took a long time again. Again, here I initiated something novel that nobody had done in the lab. So sometimes people inherit the project and then it takes much less time. But in both cases, it was not the case for me. But again, I published the paper in Science, and it was a major discovery. And I would start looking for a job. Unfortunately, it took 3 years to get a job as a PI, so I'll get into it later. So I had a really long postdoc training. Now, let me just go over briefly what we do in the lab. So autoimmune diseases are the diseases that in which our immune system attack our cells. It's very prevalent in the States and also in the world. And many of these autoimmune diseases also can be tissue specific. For example, It can happen in the joint, or in the lung, or in the pancreas, or skin, or small intestine, or large intestine, or in the brain. So it could be very specific. Target of this autoimmune disease can be very specific. So that means if you want to treat them, maybe tissue-specific suppression of immune responses would be ideal. However, in many cases, we treat the patient with systemic suppression because we don't know how to make a tissue-specific immune suppression as a treatment. So the caveat of this approach is that first, basically, if you suppress the entire system, you suppress critical immune responses in other organs without diseases. That increases the susceptibility to the lethal infection and also cancer. So the ideal way is suppressing immune responses only in a particular location. So that's why we are studying how that works. So tissue-specific migration of the lymphocyte. So peripheral tissues like gut or the skin, they secrete homing cues. They're metabolite, tissue-specific metabolite. And then they are signaling the T cell receptor, oh, you should, once you get activated, you should come back, all right? Then the T cells express the homing receptor, whose ligand is expressed on the endothelial cells of the target tissue. So once they leave the lymph node, and they just go back to the target tissue. That's how it works. So for this tissue-specific migration of lymphocyte, you need two sets of molecules. First one is chemokine and chemokine receptor, and integrin and integrin ligand. So this receptor, chemokine receptor, integrin are expressed on the cells, and the matching combination of chemokine and the integrin ligand is expressed on the endothelial surface of the target organs. So it's like a zip code. If you have the right combination, cells will get out of the blood vessel and going into the tissues. Okay, so normally T cells, for example, B cells, circulate through the body with the blood vessel, and then occasionally they stop by lymph node to see, what's going on in the peripheral tissues, because dendritic cells on the peripheral tissues, which is shown in the gut here, sometimes bring the antigens from outside through the lymphatics. And then if they meet the right T cells, they will activate. But at the same time, they also bring the metabolite from the gut, for example, here, and then educate T cells to express homing receptor for the gut, all right? And then these cells will migrate out to the blood, and if they meet the right combination of the receptor, they can attach and then come out of the blood vessel, all right? And they go into the tissue and do their job. So each step in this process, we are studying, how the T cell learn where to go and how they enter the tissues and what do they do in the gut tissues itself. So those are the major questions our lab is addressing. So in the field right now, tissue-specific homing is not, the knowledge on the tissue-specific homing is not really complete. In case of skin, which is labeled in pink, there are known homing receptor and the ligand combinations and how they are induced. So in the skin is probably the only organ get exposure to the UV, right? So the vitamin D is converted to its metabolite through the help of UV, and that became a skin-specific metabolite, signaling lymphocyte to express homing receptor for the skin. In case of small intestine, vitamin A, which is essential vitamin you have to ingest from food. So because they're absorbed from the small intestine, they have a lot more than other tissues, right? And these vitamin A can be converted to the retinoic acid, and these retinoic acid is the metabolite signaling lymphocyte to express homing receptor for the small intestine. And what I found as a postdoc was that there are separate pathways. There's a separate pathway in the large intestine, and the homing receptor for that pathway is called GPR15. That was the finding that I made when I was a postdoc, and I continued to study this pathway to see what is the ligand and what is the signaling inducing this homing receptor expression. So this GPR15 was originally found as a co-receptor for HIV and SIV. And in the original paper, people showed that the expression was high in colon and also in the spleen. So that was the initial clue that I thought that maybe it's expressed in the colon. And later I had the knock-in of the GPR15 with GFP and found that it's really enriched on the FOXP3 positive Tregs, particularly in the large intestine lamina propria. If you look at the spleen, other tissues, the expression is very low, but only in the large intestine is significant. And if you make a knockout of this, then you can see that this Treg level is decreased. So it looks like GPR15 is a homing receptor bringing lots of Tregs to the large intestine, because large intestine has lots of foreign antigens, mostly coming from microbiota. So there should be a constant suppression of unwanted immune responses. That's why you need a lot more Tregs in the large intestine. And we think GPR15 is playing a role for recruiting lots of Tregs. And also in the knockout, we confirmed that upon colitis, knockout die much faster with a lot more tissue damage, as you can see. So consistent with what we expected, that GPR15 is involved in the recruitment and homing of Tregs to the large intestine. And as a follow-up, I tried to find the ligand of the GPR15. So I sorted the large intestine endothelial cells, small intestine endothelial cells, and then high endothelial adenal from the lymph node, and tried to find what kind of proteins are specifically expressed in the large intestine endothelial cells, because probably one of those are the GPR15 ligand. And based on this RNA-seq, we found a protein called C10ORF99. It's a chromosome 10 open reading frame 99. Seems to be a ligand based on the in vitro assay and also transmigration assay with the GPR15 expressing cells. So we made a knockout mice, and when we crossed the mice to the FOXP3 and GPR15 reporter, we could observe that there's a specific reduction in GPR15 positive population, which is shown here, number 2 and number 4, in the knockout of the C1099, confirming that probably this is the real ligand. And on top of that, we overexpress GPR15, and when we mix the control cells 1-to-1 ratio and transfer, these GPR15 expressing cells migrate to the large intestine tenfold better than the control cells. This advantage is all gone if you use the knockout as a recipient, confirming that indeed C1099 is a non-redundant ligand of this receptor in the large intestine. So right now, based on this, we know this is the ligand, and we have a lot more interesting study about the role of aryl hydrocarbon receptor and the tryptophan metabolite on this part, but maybe we can talk about it later. Now, going back to my experience. All right, so let me tell you about the transition from PhD to the postdoc. So to be frank, I have to tell you, I have never thought about, or I have never experienced any other job other than doing research. I thought about quitting in the middle of PhD out of frustration and also from stress, because it takes a long time, and I make a lot of effort, but still I didn't get what I wanted. So sometimes science can be cruel. So I thought about going to medical school, and I got some advice from other people and other colleagues in the lab, and decided not to eventually. The reason was because, if you change the career, the new career is something you should like, not because you didn't like the previous career, just switch to it, okay? So you really should like that career that you're going into, first of all, and which was not the case, right? Because it's not like I want to go to medical school in the PhD, so I went to go to medical school. I don't think it was the right reason. And second, you should be really good at it because you're going to make a living out of it, right? You should be really good in addition to that you like it. So that was the problem, and I think I made the right choice not to go to medical school at the time. Because there are a lot of jobs also available right after PhD training, but I think the job, the title or the position or type of work that you can do could be different depending on how much training you got before. So if you get a postdoctoral training and look for a job, you may go to a little bit more higher level to manage the other people who are doing experiments. That's my understanding. And for the postdoc position, you have to apply for multiple labs, obviously, right? If you only apply for one lab and if it doesn't work, what's gonna happen, right? So for me, I applied 5 different labs. I got an offer from 4 different labs, and I choose one of them. So for the postdoc, it's very important because that determines what you want to study in the future, even after you finish the postdoctoral training. Because once you get a PI job, you will continue to do what you have been doing when you're a postdoc, right? So that's why I think it's very important to choose the right field that you want to work on for the postdoctoral training. So that took a long time for me to figure that out, first of all. And then you may know that the financial support for the postdoc is pretty high. Particularly NIH mandate the minimum salary. So I think including the health benefit and all of these, I think the postdoc financial support is about twice more than for students. So it's a huge burden for PI. So it's not like everybody has leftover money to support postdoc if somebody wants to come to their lab. So they have to be prepared. That means if you want to apply for a particular lab, it's very important for you to contact them as early as possible, at least a year before, all right? And the best way, I think, would be contacting them through the conference, right? It's informal contact. You just meet them in the conference and just tell them, oh, I really like your talk. I'm interested in doing a postdoc in your lab. What do you think? And you can talk over the beer. And I think that kind of informal contact would be very useful for you guys. So I strongly suggest you to think about attending conference and contacting people this way if you want to look for a postdoc position. And in my case, I chose very prestigious labs. So my graduate school lab, my PI has 20 postdocs and me as a graduate student. And for my postdoctoral lab, there were about 14 postdocs in addition to me and 1 or 2 graduate students. So, and then both of them has been Howard Hughes investigators for more than 20 years. It's a very prestigious lab, and most of the prestigious lab, they expect you to get a fellowship. So you can apply for a postdoctoral fellowship or postdoctoral grant, right? So once you start a postdoc, but they expect you to cover your salary, at least part of it. So for example, for my tenure of postdoc, I got the private foundation funding for the first 3 years to cover my salary. And then I got training grant for 2 years. And later I got a career development award just before getting a job for 2 years. So I cover about 7 years of my postdoc salary through the grant. So it's kind of important if you want to apply for a prestigious lab for postdoc position. And one thing though, it's important is that some people want to finish their project. So even after defending, they want to stay sometimes to finish up the project as a postdoc. I don't think that's a good idea because most of the funding from private foundation, they're eligible only for the first year postdoctoral fellow. So let's say if you spend 1 year in your previous lab and in the second year you go to the different lab, you became already not eligible for the most of the private foundation funding. So that's something important I think you should remember. If you want to apply for a postdoctoral fellowship, you should graduate and leave right away so that you can apply for a fellowship in the new lab. There are more opportunities, okay? And then transition from postdoc to the PI job. I didn't know that I can get a PI job at the beginning of the postdoc, frankly speaking. But like in the middle of the postdoc, I start to develop pretty solid confidence that I can probably do things on my own. So that's why I started to think about getting a PI job at the time. So it took a long time for me to build the confidence for choosing my career path. And unlike the industry job, academic job process and hiring process, this takes very long time. So for industry position, if you get an offer, you're supposed to be there like in probably 2 weeks. But in academia, it takes long. Normally, job posting occurs in summer or the fall, and for the hiring in the next fall. So it takes really a long time. So that's something you have to remember. So if you miss that season, then you have to wait another year to get the job, a posting, right? And job availability varies each year. So first year I apply, I got a fair amount of interviews. Somehow I didn't get any offers. But the second year, there were only one job available in the market for the entire year. So it was very tough. That's why I had to wait for the third year for getting a job. So it's something another thing that you have to remember. And on top of this, if you have some preference for the location of the school or in the field or the focus of the department, and you have some preference, sometimes it's really hard to find a match with the academic position. And as I mentioned, probably the reason I didn't get the PI job at the first year is mainly because I think I didn't get a funding at the time. So for PI job, I think it's very important to get a good publication, and at the same time, also the funding. So funding means there are career development awards coming from NIH and also from the private foundation. So once you publish a paper and you decide to look for a job as a PI, you're eligible for this kind of funding. And what they do is they support the remaining time before you get a job, and also after you get a job, about 3 years or 4 years. And that's really a big help for you getting a job because it shows other people in the recruitment that you can really get the funding. So most of the PI job interview is composed of two parts. First is a formal presentation about your data. And normally you have a second visit. So in both visits, you will see a lot of faculties in the department. And in the second visit, what you do is a chalk talk. Chalk talk is literally you just draw things on the blackboard by chalk. And what you do is you explain your planning of your research and plan for application of your funding. So that's where you got lots of weird questions sometimes. So it's very important for you. And the next, so what determines the outcome of the job application? I think, let's say you have a very good, research result and publication, and you have a funding. That does not guarantee that you will get a job in academia, because there are a lot of other factors which are really important, which are not really in the control of you. For example, the department may have already have a particular direction in mind, in a research direction. Okay, right now, this field is very popular. Maybe we should hire somebody in this particular field, right? If you're not doing that research, then you're out of the candidacy, right? So it's something very important, but unfortunately, it's not something you can control. And second is also related, compatibility with other people's, other faculty's research. If somehow what you do can have a very synergistic effect with other people in this department, then you will have a plus, right? But if that's not if that's not the case, for example, if there's somebody doing very similar research as you do, then it's a minus, right? Again, these kind of things you really cannot control. So I think getting a PI job is very tough in that sense. Another problem is the two-body problem. So my wife was looking for academic position at the time. She's not doing biology, by the way. So she got a job already at the Maryland when I was looking for a job. So I wanted to stay in the East Coast, and that really narrowed the the school I can apply for. So always it's very tough to get a job in the same location with your spouse. But eventually things worked out pretty well because my wife got a job nearby at school right now, so we are living together. So that's about it. But one more thing I'd like to mention I didn't say, I didn't write down here, is that doing science can be very cruel, because you're studying how nature works, right? It doesn't care your emotion, it doesn't care your stress, it doesn't care your deadline for deadline for thesis or deadline for the funding application. There could be a lot of stress, right? But let's say if experiment didn't work as you expected and it turned out that you're wrong, so that you will lose the funding or you will lose the chance of publication. The question is, can you still stand firm even with those stress and pressure? And if you cannot stand that, then science is not a good career choice for you. I've seen a couple of people, sometimes real close people, who committed scientific misconduct and eventually retracted the paper. I've seen those kind of people doing. I think the reason why they're doing it is mainly because they cannot really resist the pressure from their life, But science doesn't care, right? You really have to care about truth, and that's our job. And if you are wrong, then you just have to accept it and move on. And if you cannot do it, science is not the career they should pursue. Okay, that's all what I can say.
Thank you. Yeah, I think all as, students working in a research lab, which the majority of us are, I think we're starting to understand the pressure that comes from science and, learning how to deal with it. So I appreciate that last comment you brought up. So thank you for everyone for staying. I know we went a little over time, but me personally, I really appreciated how in-depth both of you went and the thoughtful advice and honesty. I feel like that was really helpful. You answered some of the questions I already had within your PowerPoints. I don't know if you both mind. I don't know if anyone has any pressing questions, or if not, I think we might need good. That's a good question. So do you guys mind answering one quick question? And then if anyone else has any questions, they can maybe reach out to you through email.
Not at all. Happy to answer. So should I go first?
Sure.
Yeah.
Okay. Well, for me, the most important is to have the motivation and drive. As I said, if you don't have that, I really don't want to waste my time. That's the most important for me, to see that you are actually excited about doing science and you would die for it and you can commit yourself forever.
I agree with Botond, and that can lead to the perseverance, right? That's very essential for doing science in general.
Yeah.
Look, you guys know where to find me. Please email me, come and find me. Happy to sit down with anyone and talk everything through.
Thank you guys both so much for presenting. I really enjoyed listening to both your stories. And yeah, thank you from GSA, and thank you everyone for joining. We had a pretty decent amount of participants, so I was excited to see that. I hope everyone got something from this seminar series this round. And both of you are taking rotation students, correct?
That's
Yeah,
correct.
I do.
Yes. So I know we had a decent amount of first years, so a little advertisement, right?
Yeah, that's always important aside said marketing.
Yeah. I think
Okay, Gina, thanks so much for organizing the whole thing.
Yeah.
Thank you for the opportunity.
Yeah, of course. Thank you.
Appreciate it.
Have a great
Bye,
day.
guys.
Okay,
Thank
too.
you.
Bye-bye.